Yes, this is Dr. Malini Basu from Kolkata. A very good morning to all who are present and have joined this MBSI platform for PhD lecture series. We have been organizing this for a couple of months, where the different scholars who have completed their PhD very recently have present their topics of their PhD work, and giving enlightenment to the new students so that they can have good footsteps in the future. We have with us today hello, am I audible? Hello, please someone, can you say, am I audible? Hello.
Yes, ma'am.
Okay, yeah. We have with us today Dr. Kalpana VN. She is working as an assistant professor at the Department of Microbiology, Auxilium College, Vellore. She has 6 years of teaching experience and has completed her BSc in microbiology from Auxilium College and has completed her MSc in microbiology from DKM College for Women and has obtained her PhD in the year 2020 from Vellore Institute of Technology. Her research interest includes the fields like enzymology, parasitology, microbial biotechnology, and nanobiotechnology. She has published papers of national and international journals and has been awarded with Researcher Award for publishing papers in peer-reviewed international journals for 3 consecutive years in 2016, 17, and 18. She has also received award for contribution in health awareness in EEA Conference 2021 in New Delhi. So, and she has run several projects, research projects very successfully, and is the author of a huge bunch of research articles and has contributed to book chapters. She has been very aptly organized seminars and courses and other workshops. So a very warm welcome to Dr. Kalpana. And so, madam, if you could please start your presentation. Thank you all.
Thank you, ma'am, for your warm welcome. Am I audible, ma'am?
Yes, you are audible. Yes.
Good morning, everyone. This is Dr. Kalpana VN from the Department of Microbiology, Auxilium College, Vellore. I'm here to present about a topic called nanotechnology, malaria control. So mosquitoes. Mosquitoes, if you take, it is medically important arthropod vector, and it is one of the most important dangerous human health pests. Mosquito-borne diseases remains a major source of illness and cause death worldwide, particularly in tropical and subtropical countries. Mosquitoes alone transmit diseases to more than 7 million people annually. So mosquito-borne diseases like malaria, dengue, West Nile, chikungunya, Japanese encephalitis, yellow fever. So these all remains a major source of illness and cause a death, particularly in tropical as well as in subtropical countries. Among various diseases, these diseases, malaria is a life-threatening disease which is caused by a parasite belonging to the genus Plasmodium that are mainly transmitted to the people through the bite of infected female Anopheles mosquitoes. As we all know, malaria is the fifth cause of death among infectious disease worldwide, which comes after respiratory infections, HIV/AIDS, diarrheal diseases, and tuberculosis. And it is one of the second cause of death in Africa, which comes after HIV/AIDS. This is just a disease which can be treated within 48 hours, but it can cause fatal complication if the disease and treatment gets delayed. So despite centuries of efforts, malaria continues to infect millions and kill thousands. So this is about the World Malaria Report. So you can see the status starting from 2010 to 2018. So 251 million cases in 2010, 219 million malarial cases in 2015, 227 million malarial cases in 16, 229 million malarial cases in 17, and 231 million malarial cases in 2018. So 20 million fewer cases in 2018 than when you compare the 2010. But the data for the period from 15 to 18 highlights that there is no significant progress in reducing the global malarial cases during this time frame. In 2022, the number of malarial cases and related death was significantly higher than when you compare with 2019. So the 2023 malarial report, World Malarial Report, which is recently released by World Health Organization, sheds light on alarming malarial situation in India as well as globally. So this is about the malarial situation, that is malarial status in India. So India saw a 30% reduction in cases. It accounts for nearly 0.99% of global malarial death. India reports nearly 1.4% of total malarial cases in 2022, and globally 5 million additional malarial cases were recorded in 2022, taking the total to 249 million malarial cases. The highest malarial burden was occurred in Nigeria with 26.8% of global cases and 31.1% of the death. The highest was in Pakistan, that is 2.1 million malarial cases, which reflected 2022. In 2022, India accounts for a staggering, that is 66% of malarial cases in World Health Organization. Plasmodium vivax, a protozoal parasite, contributes to almost 46% of cases in this particular region. Despite 55% reduction in cases since 2015, India remains a significant contributor to the global malarial burden. India faces challenges, including a surge in cases in 2023, which is linked to unseasonal rainfall. So the diagnosis and treatment of falciparum malarial cases continues to be a challenge for healthcare providers. To overcome these hurdles, innovative solutions, along with existing tools and strategies involving vector control, mass drug administration, and disease surveillance hold the key to solve this gigantic health problem. The government of India has set a target to end malaria by the year 2030. So as we all know about the life cycle of malaria, so the malarial life cycle occurs in both stages, in as mosquito as well as in human stages. Whenever a mosquito comes and bites in humans, it releases its sporozoid, which penetrates into the liver cells, affects the liver, and then turns into merozoites and enters into the bloodstream, where gametocytes, male cells and female cells, are released. And these gametes are being picked by the mosquito whenever it takes a blood meal from the human. In the mosquito, the life cycle follows. So in the mosquito, what happens? The gametes converts into kinate and it reaches the midgut of the mosquito, turns into oocyst, finally to sporozoid. Now the sporozoid reaches the salivary gland of the mosquito. Now it is ready to infect the other person. This is a life cycle which happens in malaria. So whenever a person experiences malaria, the person has three stages, like hot stage, cold stage, and sweating stage. Hot stage, the person will have an intense heat, dry burning skin, throbbing headache, which lasts for 2 to 6 hours. Whereas in cold stage, the person has an intense cold, vigorous shivering, which lasts for about 15 to 16 minutes. Whereas in case of sweating stage, the person experiences a profuse sweating, declining temperature, where the temperature gets exhausted and the person gets weak, which lasts for 2 to 4 hours. So the definitive host will be an Anopheles mosquito, intermediate host will be a human, and the vector which is responsible for transmitting malaria is Anopheles stephensi. The season which is common, it is July to November. So majority of those who die from malaria are infants, children, and pregnant women. So there are many different species of Plasmodium, mainly like Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, and Plasmodium bovidae. The incubation period varies from 12 days to 28 days for each and every parasite. So there are many drugs in the market which is used for malarial treatment, like chloroquine phosphate, artemisinin, mefloquine, and method. Each and every drug has its own advantages and disadvantages. And it has some specific targets too. So it has been noted that the first vaccine against Plasmodium falciparum malaria, that is RTS-SAS-01, that is muscurids, has been released, and it shows only transient protection, that too particularly in infants. A great increase in incidence of malaria mortality, that is Plasmodium falciparum, becoming resistant to commonly used anti-malarial drugs. And the Anopheles mosquitoes also becoming resistant to insecticides. So hence, malaria becomes a very tough disease to tackle. Increased drug resistance, vector management, and vaccine development are all challenging. Therefore, to control the Plasmodium as well as the vector activity, we need an inexpensive, eco-friendly, and effective anti-malarial drug, which is urgently required to solve this problem. Hence, a major research area which I chose was said to be a nanotechnology. The advancement of green nanotechnology will bring a solution for parasite as well as for vector controlling policy. So green synthesized nanoparticles may be a right hop source for mosquito control agent. The use of green process for the synthesis of nanoparticles, new and rapidly developing branch of nanotechnology. However, the knowledge of bioactivity of nanoparticles against the mosquitoes and the malarial parasites is limited. So taking into consideration, an attempt was made to combine nanotechnology field with a waste vegetable source in the name of green synthesis and planned to control both vector as well as the parasite. Among the vegetables, the family Cucurbitaceae is considered to be a major consumable food. So the efficacies of Lagenaria cicaria, which have been documented in this study. So the peel of this fruit has been discarded, which is discarded as waste when used for the study. So the reason for selecting this bottle gourd, Lagenaria cicaria, so it has various activities like it prevents constipation, treats the kidney problems, maintains temperature, has many antioxidant activity. So because of these various reasons, and based on the literature review, like since it says it has antioxidant, antimicrobial, cytotoxicity, anti-diabetic, anti-helminthic, antipyretic, anti-inflammatory, and analgesic activity. So based on the literature review, the Lagenaria cicaria have been selected for my study. So based on the above context, the present research is proposed to determine the mosquitocidal efficacy and antiplasmodial activity of eco-friendly green synthesized zinc oxide nanoparticle using this Lagenaria cicaria. Further, its mechanism and toxicity study also being carried out. To explore the study, the following objectives were taken. First, to synthesize and characterize the zinc oxide nanoparticles using an aqueous extract of Lagenaria cicaria peel. Then to determine the larvicidal, pupacidal, ovicidal, predatory efficiency and smoke toxicity of this extract and synthesized nanoparticle against malaria-causing vector Anopheles TPC. Then to determine in vitro anti-plasmodial activity of this extract and nanoparticles against Plasmodium falciparum. Then to study the mechanism of action of extract and nanoparticles on functioning of essential enzymes involved in metabolism of Anopheles stephensi larva and the inhibition of hemozoin formation in Plasmodium falciparum. To evaluate the toxicity of Lagenaria cicaria nanoparticles against HeLa cell line. So this is my overall work. So first, both the nanoparticles have been checked using, it has been checked against the vector as well as the parasite. So larvicidal, pupacidal, ovicidal, smoke toxicity and predatory efficiency were tested. And in the parasite, in vitro anti-plasmodial activity and beta hematin inhibition assay also have been done. So first, with the help of the extract, zinc oxide nanoparticles have been synthesized. After synthesized, it has been characterized by UV, FTR, SEM, METAX, XRD, zeta potential, and various characterization techniques. And then it is applied against vector as well as onto the parasite. Then finally, a toxicity study against HeLa cell line have been carried out. So coming on to the phase 1 study, I've just synthesized nanoparticles using the aqueous extract of this Lagenaria cicaria and its characterization technique. So first, the sample have been collected. It is washed thoroughly with the tap water, made into fine powder. And this powdered sample is mixed with 100 ml of the water and kept in a water bath at 60 degrees Celsius for an hour. So zinc nitrate was being used here as a substrate which has been prepared with the double distilled water. 75 ml of this zinc nitrate solution was mixed with 25 ml of the plant extract, and it has been stirred for 12 hours. The color of the filtrate was changed after 48 hours to light yellow and then to the colorless liquid. So coming on to the characterization technique, you can find a peak which shows about 385 nanometer with the optimum temperature of 80 degrees Celsius and pH of 8.5 with 120 minutes as incubation period. Coming on to FTIR, the present study reveals that the band proves the appearance of carboxylic acid in strong aromatic rings, which may be responsible for the synthesis of nanoparticles using this plant extract. And this is about the SEM. So you can find a rectangle, triangle, radial, hexagonal, rod, and spherical nanoparticles, which ranges from 20 to 75 nanometer with an average size of 49 due to the presence of zinc ions. And we have a TEM and set pattern. So you can find well dispersed and aggregates with a size ranging from 16 to 85 nanometer with an average size of 29 nanometer. And there are different sizes of nanoparticles with triangle, radial, hexagonal, and rod and rectangle shape. In case of set pattern, we can find highly crystalline nanoparticles. This is about EDAX, energy dispersive X-ray spectroscopy. Here, carbon, among the four elements, carbon occupies a high weight percentage. And we have carbon, oxygen, aluminum, and zinc. And trace amount of aluminum is also found in the zinc oxide nanoparticles with a weight of 0.37, which might be contributed by the trace aluminum impurity, which may be present in the chemicals during the synthesis process. And we have an XRD. So it's the XRD pattern of freeze-dried nanoparticles exhibited various peaks and which been calculated by using Derby-Scherer's equation. And the size was found to be 33 nanometer. The peaks were found to be very sharp. Broad peak area was observed, which clearly indicates that the high crystalline nature. Coming on to phase 2, I've just determined larvicidal, pupacidal, and bovicidal of extract and synthesized nanoparticles against malaria-causing vector Haemophilus stephensi. So first, Haemophilus stephensi larva was collected from a rice fields in stagnant water near the areas of male Basharam, and it is identified in Zonal Entomological Research Center, Vellore. To start the colony, the larvae were kept in a plastic tray which contains tap water. All the experiments were carried out at 27 degrees Celsius with 75 to 85% humidity and under 14 to 10 light and dark cycle. So the diet, like brewer's yeast, dog biscuits, and algae, in a ratio of 3:1:1 was fed for the larva. And pupa were transferred from the tray to a cup which contains the tap water. So totally 25 Haemophilus stephensi 4 insta larvae and pupa were left in a glass beaker for 24 hours containing 250 ml of water along with the zinc oxide nanoparticles with different concentration. And each test has been duplicated 5 times against the 4th insta larva and pupa. The experiment was performed under standard laboratory conditions. And Temifos was used as a positive control. Temifos is nothing but it's an insecticide which is being used as a positive control. And the mortality percentage was being calculated by using the following formula. So this is a value which have been got. So with the help of the plant extract, the mortality percentage retains to 87.6%, whereas the zinc oxide nanoparticles shows 93% of mortality. And whereas for the pupacidal, plant extract shows 89.6% of mortality, whereas zinc oxide nanoparticles shows 94.3% of mortality. And in case of ovicidal assay, eggs were exposed to various concentration of nanoparticles in peel extract of Lagenaria saccharia, which ranges from 2 to 10 ppm and 100 to 500 ppm respectively. The eggs which is disposed in distilled water serve as a control. For every treatment, 5 replicates have been retained with 20 hex for every 1 replicate. So once after completing the treatment from each concentration, the eggs were shifted to one tray having a distilled water for the purpose of hatching. The rate of hatching is calculated as 48 hours post-treatment, and the percent hatch was calculated in each instance. And this is about the ovicidal activity ratio. We have an aqueous plant extract and zinc oxide nanoparticles, so we can find the percentage of hatchability here. Coming on to the phase 3, we have an histopathological alteration and stereo microscopic analysis. So here, the larva was treated, the larva of Haemophilus stephensi was treated with aqueous extract and nanoparticles with fixed in 10% buffered formaldehyde for 24 hours. and then it is dehydrated through ethanol and xylene solutions. And the sections were mounted on a glass slide, and it is stained with amatoxylin and eosin for histopathological analysis under bright field light microscope. We can find the larval tissue damages and accumulations. So this is about the histopathological analysis. We can find a control, whereas the harrow indicates a disorder, broken epithelial cell, and a complete breakup of midgut. And we have a collapsed larval structure. So we have different concentrations here. And this is about the treatment with Lagenaria saccharia mediated nanoparticles. So here we can find a complete disintegration of abdominal region, particularly the midgut, ceca, and the whole structure have been got collapsed. And you can find loss in lateral and caudal hairs. And here you can find this picture says that there was a damage in the head, abdominal region, as well as in the thorax region and in the siphon region. And you can find some disintegration of epithelial layer of the outer cuticle. And you can find a great shrinkage in the abdominal region. The picture D, you can find a shrinkage in the abdominal region, whereas the tissues of midgut, hindgut, muscles, and nerve ganglia have been affected. Coming on to the phase 4 study, whereas mechanism of action of nanoparticles and the plant extract of the functioning of essential enzymes has been determined. So here you can find what is the mechanism of nanoparticles and plant extract on the essential enzymes which is present in the Hanophilus larva. So here, whenever if you treat this plant extract and zinc oxide nanoparticles onto the larva, so the mode of action, what happens? It goes and shrinks the abdomen. There will be a lateral hair loss and deformation of gills and deactivation of proteins are there and denaturation of enzymes, whereas DNA and RNA reduction will be there. So what happens here is the effect of enzyme level in the larval system after treatment with the plant extract and nanoparticles will give an exact understanding about the mechanism of action. And how to prepare this whole body homogeneity. Yes, sir.
7-8 मिनट हो गया.
Does somebody want to say something? I mean, you can ask your questions at the end. Otherwise, we'll lose time. So, madam, I think you should proceed.
Ma'am, shall I continue?
Yes, madam, yes.
So we have a preparation of whole body homogenates. So we can treat both the treated and untreated larva, which can be washed with the double distilled water. And the body surface have been blotted with tissue paper to remove the adhering water. And the larva can be homogenized with high cold sodium phosphate buffer. And the whole body homogenates has been centrifuged with 8,000 rpm for 20 minutes at 4 degrees Celsius. And the supernatants can be used for further analysis. So this is about quantitative analysis of biochemical constituents. We can determine the protein concentration, acetylcholine stearase assay, and carboxysterol assay. So as we all know, it's a basic assay techniques for the larva. So we need to go for, we need to take the homogenates and the respective solutions. In case of protein concentration, to detect the protein concentration, we can go for alkaline copper sulfate solution, which can be incubated at room temperature for 10 minutes. And then Folens reagents was added and it is mixed well. It is allowed to stand for 30 minutes and the absorbance, UV absorbance was read at 660 nanometer. Whereas in case of acetylcholine stearase assay, you can go for sodium phosphate buffer and can be incubated at 30 degrees Celsius for 10 minutes. And DGNB and acetyl thiocholline hydride was added and it is incubated for 5 minutes at room temperature and the absorbance was read at 400 nanometer. And whereas in carboxyl stearase assay, same to the respective homogenate, alpha or beta naphthyl acetate solution were added and it is incubated at 30 minutes at room temperature. And 500 ml of fast blue SDS reagent was added to stop the enzymatic reaction. So the absorbance was read at 588 nanometer. So this is about the results So proteins are very much necessary to build and repair tissues. So whenever the zinc oxide nanoparticle goes and attaches onto the larva, what does it do? This has a direct toxic effect on the larva, so that what happens, the protein synthetic machinery of that larva gets affected. So proteins is very much necessary to build and repair tissues and to make some hormones and other body chemicals. So there is a decrease in concentration of the protein level. And whereas in case of acetylcholine sterase, so this enzyme is very much necessary for the larva to break down the neurotransmitter acetylcholine at some synaptic cleft. So what happens here? The nerve impulse can be transported across the gap. So neurotransmitters must be cleaned immediately once the message is passed. If not, what happens? It leads to paralysis. So since here the acetylcholine sterase enzyme is getting down, it's coming down. So the larva cannot increase its nervous system. So there will be a direct toxic effect on the nervous system. And whereas in case of carboxyl sterase activity, esterase activity, these enzymes are responsible for developing a resistant mechanism in the larva. So this enzyme will cleave the carboxyl ester as well as the phosphodiester bond. So resistant insect will have some high activity of esterases. And coming on to the phase 5, that is to evaluate the predation efficiency of Coezilia reticulata, which is against Anopheles stephensi. So here what happens, I have just taken a guppy fish and it is assessed against Anopheles stephensi larva. So in each trial, 150 larvae were introduced with one Coezilia reticulata in a 250 ml glass beaker, which contains about dechlorinated water of 100 ml with different concentration. And the LC50 concentration has been taken, it has been added here. And the results shows that the experiment was performed under standard laboratory conditions without the treatment of nanoparticles and plant extract. Control was kept as dechlorinated water, the mosquito larva, without this guppy fish. So all the beakers were checked daily for 12 and 24 hours, and the number of larvae which is consumed by the fish was recorded. So here you can find the plant extract, that is zinc oxide nanoparticles and under standard laboratory condition. So the number of consumed prey, okay, total predation is the predation percentage. In case of plant extract, 45% was a predatory, and in case of nanoparticle, it is about 48.3%. And under standard conditions, it is about 44 predation efficiency. So then this is an attempt which has been made using the plant extract. Using the plant extract, I have just made an herbal cone for smoke toxicity assay against the Anopheles stephensi larva. So 4 grams of this powder, plant powder, and 2 grams of the sawdust, which acts as binding material here, and 2 grams of the charcoal powder, which acts as a burning material here, have been mixed together, and a cone has been molded. And this cone and this herbal cone has been tested for the mosquito, like huddled mosquitoes. So this is a laboratory setup. So what happens here? So fed mosquitoes and unfed mosquitoes, that is number of live mosquitoes and number of alive dead mosquitoes have been calculated. And whenever the coil is being used, you can find to 77.3% of mortality, whereas in positive control, which is being used as goodnight coil, where 93% of mortality, whereas in negative control, so it is about 11.1%. So this is about the smoke toxicity assay. And this is the paper which has been published, which is related to this article, which has been published in IET journal. And coming on to the phase 7, as of now, we were discussing about the zinc oxide nanoparticles in plant extract. Now, how does the nanoparticles works on a parasite, like in vitro antiplasmodial activity of Lagenaria cyclaria. So an evaluation of antiplasmodial activity was done against 3D7, that is chloroquine sensitive strain of Plasmodium falciparum, using a very inexpensive method, which is based on Malaria cybergreen 1 based fluorescence assay, that is MSF assay. So fresh human erythrocyte, the fresh human positive erythrocytes were suspended in a medium, and test sample plus 1% of the parasite cell suspension were taken in a 96 well plate, and it is incubated for 72 hours. Negative control serves as DMS4, and chloroquine diphosphate serve as a positive control. And 100 ml of lysis buffer have been added, and Cyber green 1 in vitrogen is a lysis buffer, which has been incubated for 1 hour at 37 degrees Celsius. Then the plate was examined at 529 nanometer using a fluorescence plate reader. And the high C50 value, that is inhibitory concentration value, have been detected, and the results were validated microscopically by using a GIMSA stain smear of both the extract and the nanoparticles. So this is about the results. The high C50 value, different concentration of the nanoparticles have been taken, and it has been acted against Plasmodium falciparum for 48 hours. And the high C50 value for the plant extract have been calculated as 14.52%. And whereas for the nanoparticles, it is 4.32%. And whereas for the chloroquine diphosphate, it is about 2.5 μg per ml. So it is being classified. It is being classified the IC50, which is lesser than 10 μg per ml, is considered very good. And from 10 to 50 μg per ml is considered to be a moderate malarial activity. And over 50 μg per ml is considered to have a very low activity. So this is a validation by using a microscopical method. So you can find the control cells. It's a normal blood cells. And we have a positive control and negative control when it is treated with Lagenaria Saicharia extract. Whereas here with the nanoparticles, you can find the Sison stages and ring stage, trophozoite, all those things have been destroyed as the concentration increases. So the parasitemia concentration.
Malaria controlled by nanoparticles, zinc oxide nanoparticles.
This is about the mechanism of malaria. This is about the mechanism of anti-malarial activity of nanoparticles through beta-hematin inhibition assay. So this is also an attempt which has been made to find out the mechanism involved in zinc oxide nanoparticles in plant extract. So first, the Plasmodium falciparum and the herbices, what happens? It degrades the hemoglobin and makes a pigment called hematin. So it is a malarial pigment. Amazoin is a malarial pigment which is present in the body. Whenever a person is infected with malaria, the person will hold hemazoin, that is malarial pigment. So when you treat with a plant extract or nanoparticles, what happens? This nanoparticles will goes and combines with the hematin so that a complex is created. Hematin plus nanoparticle complex will be created. So there will be an inhibition of hemozoin formation. Hemozoin will not be formed. That is, the malarial pigment will not be formed. It damages the DNA of the parasite and increase free heme concentration. Heme concentration has the death of the parasite will occur. So this is how the mechanism occurs when you treat a patient with nanoparticles or a plant extract through beta-hematin inhibition assay. So this assay has been carried out by using this following protocol. So different concentration of the extract and different concentration of the nanoparticles were taken, and 3 millimolar of hematin and folic acid and hydrochloric acid have been added, and the final volume was adjusted by 1 ml with sodium acetate buffer. And overnight incubation with shaker was done. During this process, chloroquine diphosphate was used as a positive control, and the samples were centrifuged at 14,000 rpm, 10 minutes at 21 degrees Celsius, and 2.5% STS and phosphate buffer saline were added to the samples in order to purify the hemazoin pellet. Then final wash is done with sodium bicarbonate until the supernatant becomes clear. Finally, the pellets were dissolved in 1 ml NaOH, and the absorbance was measured at 400 nanometer using an UV spec. So the results were recorded, and percentage of inhibition of hematin has been absorbed. It has been calculated by using the following formula. And this is about the value of percentage inhibition of hematin polymerization. So for extract, we have a 2.79% inhibitory concentration. And for the nanoparticles, we have 1.38%. And for the positive control, that is chloroquine diphosphate, we have 0.91 IC50 value. And to assess any chemical injury to erythrocytes that might be attributed towards synthesized nanoparticles was also done. This is about the next phase. So we need to check whether the nanoparticles will affect our erythrocytes. If it is taken inside the body, whether the nanoparticles is affecting our erythrocytes or not. So this is an attempt to check whether any injury is happening to our erythrocytes if we take this nanoparticles. So 200 ml of erythrocytes was incubated with 100 μg of the nanoparticles at a dose equal to highest which is used in antiplasmodial assay. So whatever the dosage which I have used in antiplasmodial activity, equivalent to that or higher to that, the dose has been used to check the erythrocytes damage. The conditions of the experiment were maintained as in the case of the antiplasmodial assay. So 48 hours of incubation was done, and a thin blood smear was stained with Giemsa stain and observed for morphological changes under high power light microscopy. And this morphological findings were compared with erythrocytes which were uninfected or which is not exposed to the nanoparticles. So you can find different concentration of erythrocytes which has been treated with plant extract and nanoparticles. So this shows you can find there is no morphological differences after 48 hours of incubation. Even though after 48 hours of incubation, when it is treated with plant extract or nanoparticles, it doesn't show any effect. And the last phase is about to check the toxicity of the nanoparticles towards a HeRA cell line. Just check. I've checked how toxicity it is through a cell line. So different cells were placed in a 24 well plate and it is incubated at 37 degrees Celsius with 5% carbon dioxide condition. Once the cell reaches the confluence stage, various concentration of the sample were added and it is incubated for 24 hours. After incubation, the sample was being removed from the well and washed with a phosphate buffered saline. And MTT assay was done, and 1 ml of DMSO was added to all the wells, and the absorbance was measured at 570 nanometer using an UV spec. And IC50 value, 50% of inhibition value was being determined graphically. So I've used different concentrations here and different dilutions and absorbance and cell viability percentage was carried out. So 62.5 μg per ml shows a 50% of value, cell viability value. So as the concentration decreases, you can find the cell viability increases. So this shows that the nanoparticles is dose dependent. It is not toxic, but as the dosage increases, it may reach in toxic. And this is the normal HeLa cell line. Whereas in case of 1000 μg per ml, you can find the cells are getting damaged. Whereas in 17% of the cells are viable, whereas in 62.5 μg per ml, you can find 50% of the cells were viable, and 7.8 μg per ml of ml, you can find 80% of the cells are viable. So as the concentration increases, cell viability also decreases. So to conclude, while social distancing and a national lockdown have been initiated by the government against COVID-19 pandemic, there are occasional bouts of disease like malaria, dengue, and yellow fever, which requires equal attention. March and April are crucial months when preventive measures like fogging, fumigation, awareness campaigns are undertaken. So currently, this work is halted due to a nationwide lockdown. The peak season for malaria starts from May and a spike is seen after the monsoon in July and August. So it is an apprehension that malaria prevention might take a backseat since resources are being directed towards COVID-19. But we need to ensure that does not happen. Spreading awareness about malaria and preventive measures are very important. So overall, this research highlights that aqueous extract of the plant extract and its mediated nanoparticles can be employed at very low concentration to control the Anopheles stephensi population. So in this study, the nanoparticles were green synthesized using an aqueous extract, and this acts as a reducing and capping agent for Lagenaria cicaria. The mechanism of action was investigated on larvae, adding knowledge on how the nanoparticles affects the mosquito cellular physiology. So the present investigation also shows that this plant extract and its mediated nanoparticles have been found to inhibit the growth of falciparum in human red blood cell culture. So moreover, this extract-based smoke repellents may be cheaper and safer if you compare with commercial-based coils currently marketed. So green synthesized nanoparticles are environmentally safe and can be used to develop new agents for vector as well as for parasite control. Further research on employing nanoparticles on mosquito breeding fields could bring a very promising target drug which can be used for protecting malaria. So the outcome of this study will be a breakthrough in the area of malaria research. It may be a strong alternate to current anti-malarial drugs and drug combinations to combat with the drug-resistant parasite. So though it is a premature to conclude at this stage that herbal nano combinations can be used as effective anti-malarial, this finding will provide some foundation for further exploration of new effective herbal compound with nanoparticles for protection from malarial parasite. Thus, the present study will benefit the society by reducing the morbidity and mortality which is caused by malaria. So the baseline data obtained will help in recommending a new drug in future. So these are the publications which is related to the work. And these are other publications. These are the book chapters which have been published. These are the conferences which I have attended. These are few references. So to acknowledge, first and foremost, I take this opportunity to express my sincere gratitude to my guide, Dr. V. Devi Rajeshwari, Assistant Professor Senior, SBST VIT, Velu. Without her motivation and continuous encouragement, this research work would not have been successfully completed. Also grateful to our Chancellor for motivating me to carry out research in VIT and also for providing me with infrastructure facilities and many other resources needed for my research. I express my profound gratitude to my doctoral committee members, Dr. Nandini Devi ma'am, Anna University, Dr. Vijayan Shiva sir, Madras University, and internal member, Dr. Jayaraman sir, Dean Academics VIT, to complete this research with their valuable suggestions. I wish to extend my profound sound of gratitude to Dr. Malini Basu ma'am, convener, MBSI, and chief patron, Dr. Arvind Deshmukh sir, for giving me this opportunity to present my research work in an open forum. Thank you all.
Thanks a lot, Dr. Kalpana. Like, this is a very clear and promotional piece of work. So you have done a very sequential mode of studies, which is definitely very benefiting, not only to provide directions, but having an immense societal impact. So I hope like all these, like eventually you will work more on in this direction and like have some patenting and things like that, which are like the theme of the day today, like everywhere is talking about that. I think if participants have questions, you can just unmute yourself and ask to the speaker, and she will be more than happy to answer you. And I would like to make one announcement, and it's a request on part of NBSI. Please mark your calendar and the time of the day to enter the meeting, because in between, like entering and not only almost close to the session, I found lot of participants entering. So it is not a very decent activity, and not only that, it also is not very good on our part, because then we get distracted and then speakers also they lose their concentration. So please make it a point that you attend the meeting on time, which will be beneficial both for your end and our end. So questions, please, if participants, if you have it. I guess nobody has so, but I myself would like to ask one, like you made the aqueous extract. So is the most optimal thing, or like you can make a extract in any other liquid also?
It is not only using aqueous extract. We can go for various solvents also. Like ethanol, methanol, so various solvents also we can use. But to simplify the work, I have been used this aqueous extract, and that's why to make the process more simpler and more way. My work is to do it in nanoparticles. So this is just to compare. The aqueous extract is just to compare with the nanoparticles.
Okay, so keeping water as a baseline actually.
Yes, ma'am.
Yeah, and other thing like for treating the larva, etc., etc. So you have collaborations with like zoology or something, or like you all did it. You have your setup only.
We have a setup. We have a setup, ma'am, in VIT. There is a lab which is mainly for larval cultivation of larval.
Okay.
So we did it there.
Okay. Okay. Great. So thank you all once again. And next week we might not have the session for some issues. But the week after, we will definitely have it. That is on the 23rd. And at that time, Dr. Kirat Ganguly, sir, he will be conducting the session. So please keep an eye to the notices and the posts in the Microbiology Society group so that you get aware of the topic and the speaker. Okay. Thank you all once again. Have a nice day.
Thank you, ma'am.
Sure.